So he got to ask his question, right? Okay, so the last speaker of the session is James Surmeier. He's the chairman of the Department of Physiology at Northwestern University in Chicago. And his lab looks at how neuromodulatory systems shape the excitability of basal ganglia and frontal cortex neurons. This is a highly complex problem because obviously there's a high molecular and circuit diversity in these systems. And so to overcome these obstacles, he uses, his lab with him obviously, uses a combination of electrophysiological, biochemical, and molecular strategies together with really state-of-the-art single-cell RNA-seq and other approaches to really tackle how what are the exact mechanisms by which these neuromodulatory systems impinge on these cognitive systems. So we're really looking forward to your talk, and welcome, please, with me, James Surmeier.
Well, I want to thank the organizers for the invitation. This is my first visit to Lisbon. It's a beautiful city, and what an incredible facility this is. I am really very, very impressed by it, and looking forward to the rest of the meeting here to learn more about what's going on here and to learn from the rest of the participants in this symposium. Now, one of the things I have to say, though, is when I started and looking at the program, I thought, did I come to the wrong meeting? Because I really study Parkinson's disease, not so much Alzheimer's disease, but Rui has assured me that I did come to the right meeting, and so maybe you can decide after I finish. So I'm going to tell a simple story today, actually. I'm not going to talk about the work that was mentioned in the introduction. A lot of the work that we do is focused on basal ganglion circuitry and how that's altered in disease states like Parkinson's and Huntington's disease. What I'm going to do today is I'm going to talk a little bit about pathogenesis in Parkinson's disease, the second most common neurodegenerative disease. And I think we can learn some lessons about Alzheimer's disease from studying Parkinson's disease. I think there are common elements to the two diseases. We know that there's no cure, like for Alzheimer's disease, there's no cure for PD. And so if we're going to develop a way of slowing the progression of the disease, we need to know something about how it arises, what causes it. And of course, there are lots of people thinking about this. We've heard a lot of ideas about pathogenesis. Certainly age is the #1 risk factor for both AD and PD. Genes and environmental toxins clearly have been implicated in PD pathogenesis. The thing that those things do not explain is the very distinctive pattern of pathology that you see in the human suffering from Parkinson's disease. Now, one of the hallmarks of PD pathology is Lewy pathology, Lewy bodies, which are shown here. This may be, is this from one of your papers? No? Now, Lewy pathology in clinical PD is distributed, and it is commonly correlated with disease progression. So in the early stages of the disease, you see it primarily in the caudal medulla. In the middle stages of the disease, where patients become symptomatic, it's more distributed. It seems to move more rostrally. And in the later stages of the disease, it's found in higher density in the medulla and also in telencephalic structures. This gives the appearance that the disease pathology spreads through the brain. Okay? Now, one of the caveats here is that the distribution of pathology in all of these stages, in spite of the fact the way it's drawn, is still discrete. The brain does not fill up with Lewy pathology. It's not a gas tank. The pathology is discretely localized, and it's in a small percentage of the neurons in each one of these loci. Moreover, the pattern of Lewy pathology is variable. Only about half of the PD cases, clinical PD cases, conform to this sort of Brock-like staging. So it's widely assumed, though, that misfolded alpha-synuclein spreads through the brain connectome in a prion-like fashion, causing that pattern of pathology that I just showed you, which then is hypothesized to lead to dysfunction and neuronal death. Okay? Now, this is important to me because it makes vulnerable neurons simply bystanders. They don't participate, really, other than getting in the way of the prion train. So one very simple question is whether or not that model is consistent with the connectome of the brain. Now, detailed connectomes have not been generated for many parts of the brain. I'm just going to show you one, where we have monosynaptic rabies virus tracing techniques where we can quantitatively map inputs to a structure that we know exhibits Lewy pathology to see whether or not the pattern of Lewy pathology spread from that point is consistent with that connectome. That is, are cells that make strong connections with that source of Lewy pathology more likely to manifest Lewy pathology than regions that don't have connections or have very weak connections. This is a diagram constructed. Here's the locus coeruleus. This is the seeding site. The size of the circles are the extent of the innervation of the locus coeruleus. Cells here have Lewy pathology. Cells here don't have Lewy pathology. And what you can see from this map is there is no correlation between connectivity and the probability or extent of Lewy pathology. Particularly interestingly, GABAergic neurons seem very resistant to manifesting Lewy pathology. So this does not argue that there's not spread, but it argues that something else affects the degree of spread of Lewy pathology in the Parkinsonian brain. The other thing that I think is really important is to look at the pattern of cell death. Cell death is clearly correlated with symptoms. Lewy pathology is not so clearly correlated with symptoms. The earliest cell death in Parkinson's disease is in the substantia nigra, right, pars compacta. And later, it begins to appear in the pedunculopontine nucleus, in the intradolaminar nuclei of the thalamus, and then begins to appear in the caudal medulla, where the Lewy pathology starts. So there's a dissociation between cell death, which is clearly linked to symptoms, and Lewy pathology, which again suggests to me that there are some cell autonomous factors that are involved in pathogenesis. So that's what I want to spend the rest of my time talking about. The first neurons to die are dopaminergic neurons in the substantia nigra. This is a pigmented nigra. This is the nigra of a PD patient that has lost those neurons. What might make those cells vulnerable? And again, I'm going to skip over a lot of work, summarize a lot of work here very quickly. These cells have a very distinctive physiological phenotype. So in keeping with Liwei's idea that physiology somehow has an impact on pathogenesis, these neurons are all autonomous pacemakers. They have very broad action potentials. They're just like cardiac cells. They beat at 2 to 3 to 10 times a second all the time. Now that pacemaking is necessary to maintain dopamine levels in target structures like the striatum that control action, selection, and habits. Okay? Stuff that Rui has spent a lot of time characterizing. Those neurons must also increase their discharge rate in response to stressful conditions where, for example, the one situations that we've encountered, maybe not so much now, but certainly in our distant evolutionary past. So they have to be able to maintain that basal discharge rate and accelerate their discharge rate and keep that going for a very long period of time. Okay. So the way that these neurons are designed is that they have redundant pacemaking mechanisms. They have an axon initial segment pacemaking mechanism that depends upon sodium. They also have an oscillatory mechanism in the dendrites which depends upon calcium. CAV1 channels, L-type calcium channels in particular. And those two pacemaking mechanisms are redundant. They oscillate at almost exactly the same frequency. They support each other and make pacemaking much more robust, and any kind of activity-driven pattern more robust. Now, this oscillatory redundancy is peculiar to compacted neurons. It's not seen in ventral tegmental area neurons, dopaminergic neurons, which are neighboring, which are not so sensitive in PD. Okay. So a very fundamental question associated with maintaining this high level of activity and these redundant pacemaking mechanisms is how these neurons meet the bioenergetic needs associated with that. After all, every time a cell fires a spike, it dissipates ionic gradients which have to be restored through ATP-dependent pumps and exchangers. So spiking electrical activity is bioenergetically very expensive. We all know that mitochondria are the principal powerhouses of neurons, right? So the burden undoubtedly falls upon their shoulders. it looks as though, and I'm summarizing a lot of work here, that compacted neurons are a lot like muscle. Muscle uses a feed-forward system to control mitochondrial respiration. When calcium is released from the SR to drive the contractile machinery, it in parallel is injected into the mitochondria to accelerate the production of reducing equivalents for the electron transport chain. So this is a sort of a cartoon summary of this. This oscillation that's happening during the pacemaking opens these channels, which drives calcium-induced calcium release from the ER, this is again very, very important, drives calcium at specialized junctions called mitochondrial associated membrane into mitochondria, which disinhibits the TCA cycle, which drives oxidative phosphorylation. Okay? So this is a feed-forward mechanism that helps accelerate the production of ATP in anticipation of need. Okay? Right? Because I think this is very, very important. This is part of again, the design feature of muscle. It's found in these cells and I think a variety of other neurons that are vulnerable to aging-related diseases. Again, CICR is a big part of this, and I think one of the unappreciated features of CICR is its regulation of bioenergetics. Now, again, I'm not going to show you all the data. I'm going to show you a little bit of data. One of the things we've done is we've monitored calcium entry into the mitochondria using a genetically encoded probe. We can express this just in dopaminergic neurons. And one of the things we can see, again, here's the probe in mitochondria in a dopaminergic neuron in an ex vivo brain slice, is mitochondrial matrix calcium tracks spike rate. This is just a plot of spike frequency as a function of mitochondrial calcium. So the mitochondria are keeping track of how much the cell is spiking and regulating oxidative phosphorylation in parallel. Now, one of the ways we can gauge the importance of this for the functioning of these cells is to sort of mimic this situation, this cartoonish situation. We could do that by elevating extracellular potassium, which drives the activity of compacted neurons. And normally, they don't have any trouble maintaining that discharge rate. And as a consequence, you shouldn't have any trouble maintaining the selection of appropriate actions to escape a predator. If we take away the L-type calcium channels to eliminate that bioenergetic control mechanism, what happens is when we elevate activity after a little period of time, the dopaminergic neurons collapse, they stop spiking, dopamine levels in the striatum fall, you become Parkinsonian, and you don't escape. So there's been strong evolutionary pressure, I think, to maintain this sort of design because it affords a survival advantage for us and all the other animals that use the same sort of basal ganglia structure. The trouble with the design is it comes at a cost, and that is the production of reactive oxygen species. Driving oxidative phosphorylation clearly increases the production of ROS. Now, we can see this, again, in ex vivo brain slices of compacta by measuring with a mitochondrially encoded redox sensor, or targeted redox sensor, oxidant stress. In a ventral tegmental area dopaminergic neuron that's pacemaking, there's no oxidant stress. In a compacted neuron, there clearly is elevated oxidant stress. That oxidant stress can be diminished by diminishing calcium entry through L-type calcium channels and decreasing the calcium loading of mitochondria that slows oxidative phosphorylation in the mitochondria. So we know that oxidative stress is implicated in a wide variety of cellular dysfunctions associated with aging and neurodegeneration. Leads to increased mitochondrial DNA deletions, leads to telomere shortening, leads to DNA mutations, and also leads to deficits in proteostasis. All the things that we know are important in both PD and AD. So one of the questions, though, is whether or not the oxidant stress that we see in our experimental models is actually sufficient to cause damage in vivo. It's good in principle, but is it good in practice? And so one of the ways we've attacked this is by using mitochondrial turnover or mitophagy as a measure of damage associated with oxidant stress. And we use a probe called mitokima, which allows us to measure mitochondrial turnover in vivo. This probe changes its excitation spectrum in response to changes in the pH of the environment. So the bottom line here is that what we can see is in ventral tegmental area dopaminergic neurons, the mitochondria are typically not in lysosomes. A small fraction of them are in lysosomes when we do a cumulative map over the course of a week. But in compacted neurons, there clearly is a much higher fraction of the mitochondria that have migrated into the lysosomal compartment as a consequence, we think, of this oxidant stress. Okay, so the question is, we don't live on the savanna anymore. Can we re-engineer these dopaminergic neurons to be less stressful, to slow their loss with aging? So the idea here is that we have drugs that are very good antagonists or inhibitors of CIV1 channels, the channels that are in the plasma membrane that have been implicated in the bioenergetic control mechanism I talked about. So what we did is we chronically treated animals at a clinically relevant dose for about a week, and then went and looked at the tissue. We saw 4 effects. One is cytosolic calcium levels. These are just 2-photon laser scanning microscopy images of dendritic calcium oscillations during pacemaking. There's an electrode here. Calcium levels go down. I was really concerned about that because we know that with sustained inhibition of a lot of proteins, the cells will upregulate that protein to compensate. This doesn't happen in this particular case. L-type calcium channels stay low. The other thing that happens is mitochondrial stress drops. It drops more than it drops with acute isradipine treatment. So this is just chronic stress. This is acute treatment. This is the controls. The other thing that's very interesting is mitophagy rate slow. This is the data that I showed you. This is a VTA neuron. This is a compacted neuron where we're looking at a mitophagy over the course of a week. After a treatment with an L-type calcium channel inhibitor, the mitophagy rates in compacted neurons become like VTA rates. You're dramatically slowing mitophagy and diminishing the proteostatic stress that these cells are constantly undergoing. And lastly, consistent with the whole idea that you're decreasing mitophagy, mitochondrial mass rises in compacted neurons. This is an example of a mitochondrial label in a compacted neuron from a mouse ex vivo brain slice. This just shows you the nucleus and the cell body, or the somatic area that's stained with tyrosine hydroxylase. This is after treatment with isradipine. You can clearly see the difference in the amount of green in those two. And 3-dimensional reconstructions of the mitochondrial network in these cells has shown that mitochondrial mass rises by about 30%. Okay. Everything I've told you up to this point is from mice. Mice don't get Parkinson's disease. We don't have a good model of Parkinson's disease that has predictive validity for humans. All right, so why should we believe that this applies to humans? Let me give you a couple reasons. One is, if you look at postmortem brains and you look at mitochondrial DNA in the compacta as the most vulnerable region, what you see, and everybody who's done this has seen the same thing, you see a high level of mitochondrial DNA deletions, which is typical of regions that have high mitochondrial oxidant stress. So in humans, compacted neurons have high mitochondrial oxidant stress just as they do in the mice. All right, so that maps. But probably the most convincing evidence, and I'm not going to go through all the human evidence, these are the most important things as far as I'm concerned. Humans have been taking dihydropyridines for the treatment of cardiac hypertension for a long time. CIV1 channels are found throughout the cardiovascular system. And so inhibiting them relaxes vascular smooth muscle, lowers blood pressure. If you were treated for hypertension with dihydropyridines, but not ACE inhibitors or beta blockers, which are actually more effective, right? But if you're treated with dihydropyridines, your risk of developing Parkinson's disease drops by 30%. Which to me is astonishing given the fact that most of those patients were taking extremely low doses and taking dihydropyridines often that didn't have good blood-brain barrier penetration. Okay. So, I'm on time. I was worried about that. No? Okay. The truth of the matter is, though, the only way to really know is to do the human experiment. Human experiments are expensive, time-consuming, but about 5 years ago, NIH bit the bullet on this in the United States and funded a 5-year, not a 1-year or a 2-year, but a 5-year clinical trial. It's $23 million. There are 56 centers. Those centers are shown here all over the United States and Canada. And the reason there were so many centers is we wanted to get patients at the very early stages of the disease, just after they were diagnosed. They couldn't be on any symptomatic medication, right? So we're trying to capture them as early on in the disease, evolution of the disease as possible. There were about 350 patients that were enrolled. Enrollment was achieved very early. The study results will be announced early next year. So we'll know. Okay, so let me summarize what I've tried to tell you in the next 23 seconds. I've tried to convince you that vulnerable substantia nigra pars compacta neurons have an unusual physiological phenotype that's demanded by their role in controlling movement. There have been strong evolutionary pressures to shape this physiological phenotype. I think it's a phenotype that is shared by many other of the neurons that die in Parkinson's disease. Okay? Again, the data set's not complete, but I think that it's a reasonable inference. It's certainly true of locus coeruleus. It's certainly true of dorsomotor nucleus of the vagus. A key part of that phenotype is the engagement of CAV1 or L-type calcium channels to help meet the bioenergetic needs of the cell in times of fight or flight. They have to stimulate oxidative phosphorylation to keep the cells from shutting down during times of stress. Again, remember, if ATP levels drop, KTP channels get activated, neurons shut off. Okay? That's a protective mechanism that all neurons have. But that design comes at a cost, and that is increased mitochondrial oxidant stress. In the short term, this is not a problem. Right? It's only over very long periods of time, decades and decades, that the deficits associated with that mitochondrial oxidant stress begin to accumulate. Average age of diagnosis with PD is 60. There couldn't have been no evolutionary pressure to get rid of this design feature, because you're well past reproductive age at that point. Now, we know that antagonism of L-type calcium channels lowers oxidant stress. That is systemic treatment with L-type calcium channels lowers oxidant stress. There are no other deleterious side effects that we know of. Again, it's been used for 20 years in humans. It decreases mitophagy, decreases the perostatic burden associated with Lewy pathology or alpha-synuclein transmission, which may very well be happening. It also increases mitochondrial mass. I also want to emphasize one of the things that we know increases aggregation of alpha-synuclein are reactive oxygen species. Now, in humans, we know that antagonizing CAV1 channels is clearly associated with decreased risk. There's also evidence that it is associated with decreased progression of the disease, even if treatment onset is late. And lastly, we'll know very soon, a phase 3 clinical trial was mounted and is near completion in the States. So we'll know whether this is the first neuroprotective agent for Parkinson's disease. Okay, so let me tell you who actually did this work. This always takes a village, right? I mean, there are lots of very talented postdocs. I didn't do any of this work. I just sit on the sidelines and cheer. Jaime Guzman, Emma Illich, Ben Yang, Enrico Zampizzi, Patricia Rodriguez, a new addition to the lab from Spain, Dan Galtieri, David Wilkeson, Zhongzi, Kristen Stout, Savio Chan, Dillian Drojanowski, and Javier Sanchez all made seminal contributions to the work that I showed you. I also have strong collaborators. Paul Schumacher developed the mitoRO GFP mouse that allowed us to, for the very first time, to see in vivo and in intact brain slices mitochondrial oxidant stress in a behaving cell, neuron, a real neuron. Tanya Samuni headed the phase 3 clinical trial. Glenda Halliday and Jose Obiso have taken me to school on the clinical aspects of PD. I think it's extremely important for basic scientists to talk to clinicians on a regular basis. They helped shape my thinking and focus our research effort. Jose Lopez Barnio is collaborating with us on work that I didn't show you, looking at the effects of long-term deletion of electron transport chain proteins. Serena Rizzuto and Tullio Bazan at the University of Padua have given us the mitochondrial calcium probe. And of course, none of this would have been possible without financial support. So at that point, I'll stop and take any questions you might have. Thanks.